My name is Daniel Lubicki. I am an undergrad at UMass Amherst working in the Hebert Lab, and today I'll be talking about my honors thesis project, which is the utilization of N-glycoproteomics in human cell lines to identify N-glycosites. N-glycosylation is a common protein modification. Around 7,000 proteins, which is a third of the human proteome, is targeted to the secretory pathway. Most of these proteins receiving an N-glycan upon entry into the ER. Either co-translationally with this OSTA complex or post-translationally with this OSTB protein complex. N-glycans are added en bloc. The primary N-glycan, which is a branch carbohydrate added to a protein, is 2 N-acetylglucosamines, 9 mannoses, and 3 glucoses. And this whole structure is added all at once to this consensus motif, which is N, X, T, S, and less frequently C. X being any amino acid which isn't proline. There are also other consensus motifs and non-consensus binding, but this is the primary one. N-glycans assist in protein function and ligand substrate binding and are essential in protein solubility. They are very versatile and essential modifications, especially in the ER. Based on the structure of the glycan, it can recruit different lectin chaperones, which allow it to help the protein fold. Glycans are very essential in the ER folding and quality control systems. So my goals for this project are first to identify the N-glycosylation sites on the ER resident TTC17 due to ongoing lab work within my lab, and then also map and compare N-glycosylation sites in different human cell lines. So TTC17 here, this is the putative topology and features. It consists of a signal sequence which targets it to the ER, 12 N-glycans, plenty of cysteines, and multiple different TPR domains. TPR domain is a protein-to-protein interaction motif. TTC17 is highly glycosylated. Using a PNJS-F assay, PNJS-F is a glycosidase which cuts any N-glycan right at the primary N-acetylglucosamine. And as we see, there is a large size shift here between the wild-type TTC17 and the deglycosylated TTC17. And using different pre-stain ladders as standards, we were able to create a function determining the size shift between the deglycosylated 17 and due to gel motility. And there's about a 26 kilodalton size shift. And TTC17 being an ER resident most likely has high mannose glycans, seen here to the left, one of these structures and anything in between. So we estimate around 1.8 kilodaltons per N-glycans, which leaves us with about 12 to 14 occupied sites on TTC17. So most of those putative sites, if not all, should be occupied. Deglycosylation via PNJS-F creates this scarring motif via deamidation reaction, which changes an asparagine to an aspartic acid. And this can be seen and identified via mass spec analysis. So the further query and exactly identify the TTC17 and glycosylation sites, we created a method for mass spec analysis. First thing we do is you overexpress TTC17 with a flag tag and isolate that protein, quality check on an amino blot, trypsinize the sample, deglycosylate via PNJS-F, and then send it for mass spec analysis. Using this method, 2 sites were identified. This first one here and the second one here, which is interestingly enough is within the TPR domain. A few other sites have been identified by other lab work within the lab and this glycoproteomics reference. The glycoproteome of cell lines is the next step of my project. We are going to look at HEC, which is kidney cells, H4, which is neuroglioma cells, U7, which is liver cells, and HeLa, which is ovarian cancer. The first thing we did is a Con-A affinity purification. Con-A is a lectin, which binds to high mannose glycans. And we looked at HEC, H4, and U7 cells, and we see differential banding, slight differential banding between cell lines, suggesting that there's differences in the high mannose glycoproteomes of these cells. We developed a method to further analyze the glycoproteomes of cell lines using these 2 papers, which map N-glycosylation sites across a multitude of tissues and species. The first thing we did was to isolate the secretory pathway using this method that was developed in the lab. By mechanical homogenization and differential spinning, we can isolate the endomembrane compartments, further enriching the secretory pathway where N-glycosylation occurs. And then we add trypsin and use the FAST method, which was developed in those two papers I talked about previously. Using 30 kilodalton filters, we are able to isolate N-glycosylated peptides using lectins, which bind to the glycans. We use multitudes of different lectins to isolate a multitude of different glycoforms. Then PNGSF is added to free the peptides from the lectins and also create the scarring motif, which can be identified via mass spec. Using this, we have identified N-glycopeptides in H4 cells, with over 2,000 proteins and 40% of those being glycoproteins. Data for Q7 and HeLa has not been done yet, and we're still waiting for those results. This could reveal modification changes based on tissue, or if it is conserved throughout the human body. And this maps the N-glycoproteome for other researchers. So the future directions using this method should continue to map sites in different tissues, test how N-glycosylation is affected when the ER is under stress, and test it in Alzheimer's cells versus healthy cells, as there is evidence in Alzheimer's that the secretory pathway and N-glycosylation is affected.